Welcome back. So what we're going to look at now is some specific mechanisms of how we execute mitosis. And so let's just put that in the context of the big picture. Remember, we've got 4 stages to the cell cycle. We've got gap 1 phase, followed by S phase when we do DNA replication. Following that, we have gap 2 or G2 phase, and then we go into M phase. And then M phase, remember, is broken down into the stages of mitosis. So those are prophase, prometaphase, metaphase, anaphase, and telophase, and that is followed then by cytokinesis. So mitosis and cytokinesis make up this stage called M phase. So we're going to look specifically at mitosis to start with, and then we'll finish up with looking at cytokinesis. So we're going to break this down and look at different stages. So let's start off over here on the left. Let me get a pen so we've got some drawing abilities. Okay, so let's start off with just interphase. Now, I've not really spoken about interphase. So interphase is are the stages of the cell cycle, which are G1, S, and G2. So if we're talking about interphase, the cell could be in any one of those stages. So that's just a collective name for G1, S, and G2. So is what we're going to look at in each of these stages of mitosis once we've kind of done interphase here, is we will look at what the DNA is doing, so the chromosomes and what they're doing. We're going to look at the nuclear envelope, and we're going to look at the microtubules, and we'll look at some structures called the centrioles, or maybe we just call them centrosomes, which are composed of centrioles. So over on the left here, you can, at the top panel, you can see a cell, and here is its nucleus, and the nucleus has been stained in blue. So it's a blue dye that binds to the DNA, and then you can visualize that with a special type of microscopy. The microtubules have been stained in green, and the actin filaments in the cytoskeleton have been stained in red. Now, when you have the green on top of the red, so when you see the microtubules on top of the actin cytoskeleton, when the green is on top of the red, you get yellow. So sometimes you're going to see yellow. So all of these kind of fibers that you can see here kind of radiating out. Those are the microtubules, and then you can see kind of also some actin as well. So in interphase, so what we have going on is we have an intact nuclear envelope, and we've got the chromosomes, and they're all kind of, they're not condensed, they're kind of stringy like a ball of wool if you want to think about it like that. And then we've got some duplicated centrosomes, and so those centrosomes are going to become really important when we go into mitosis. So let's do that now. Let's go into the first stage of mitosis, and let's talk about what happens in prophase. So this is the first stage. So the first thing that's going to start happening is that the chromosomes start to condense down. So this is the DNA becoming more compacted, so you have very discrete chromosomes. And so you can see the chromosomes and the pairs of sister chromatids in the nucleus at this point. And so here's the nuclear envelope. And then if we look at the microscopy image at the top, you can still see our nucleus kind of here that I'm outlining in red. You can start to see the chromosomes, actually, you can actually start to see them forming more discrete condensed structures. So the other thing that happens here in prophase are these structures called the centrosomes, they separate, and so they actually push one another away using microtubules. So the microtubules originate from the centrosomes, and they overlap and they start to push the 2 centrosomes away. So I'll just put 2 little dots on the centrosomes. There's one, well, little dot. There's one there, and there's one there, and you can see all of these microtubules radiating out from the centrosomes there. And those are going to become really important in a moment. So that is prophase. Let's go now into prometaphase here on the right-hand side. And so in prometaphase, the chromosomes now become really condensed. And so you can see these really distinctive chromosomes that are starting to form in the cell. And so they've gone from being kind of stringy kind of structures to much, much more condensed. And you have to do that because you can't pull 2 long, stringy pieces of spaghetti apart very easily, if you think. And so the cell condenses them down so they're much shorter and stubbier, and that makes it easier to separate them when we do the rest of mitosis in a moment. The other thing that's going to happen here in prometaphase is the nuclear envelope is going to break down, and so the nuclear envelope breaks down so that the chromosomes can be moved to opposite ends of the cell. So in this diagram down here, you can't really see the nuclear envelope. It's been broken up into small pieces, and then later they'll all come back together to form new nuclear envelopes. The other thing that's happening is you can see that our centrosomes have now got to opposite ends of the cell. So they started separating in prophase, and they've pushed each other away, and now we start to get these these microtubules which radiate out and start to attach to the chromosomes, and they attach to the chromosomes at points called the kinetochores. So these yellow dots in the middle of the chromosomes are called kinetochores, and so the microtubules that radiate out from the centrosomes attach to the kinetochores. And so there's this kind of dance going on with these spindle fibers. I guess it's called the mitotic spindle. These fibers, these microtubules coming out from both ends and attaching to the chromosomes. And you can see that in the top here, where you can see the chromosomes all kind of starting to kind of project out into the middle of the cell there from the centrosomes. So that is pro-metaphase. Let's go on to look at metaphase, which is our next step, and that is over here on the left side of the slide. So we're in metaphase here. Now metaphase is very, very, very distinctive if you look at this down a microscope. So what's happened here is all of the chromosomes are now being attached to the ends of microtubules, and they've all been pushed and pulled until they get to the middle of the cell. So there is a structure in the middle of the cell here which kind of runs like this from bottom left to top right, called the metaphase plate. And that is, it's not actually a structure, but it's just where all the chromosomes line up. So all the chromosomes have lined up, and you can see that all of the microtubules that are coming from the left here, they're going to be attached to one of the sister chromatids, and the chromosomes from the right are going to be attached to the other sister chromatids, and all of these chromosomes are now arranged in the middle of the cell down this structure called the metaphase plate. So that's metaphase, super easy to see. Let's now look at anaphase. Now in anaphase, what's going to happen is the microtubules start to shorten. They start to shorten and the cell gets longer, and so the centrosomes move apart from one another. So the centrosomes are pushed apart from one another. So you've got to imagine, if this is kind of, my fingers are like the spindle, you've got to imagine two things happening. One is that the spindle elongates and it pushes itself apart, and so the cell gets longer. And the other thing that happens is that the microtubules start to shorten, and as they shorten, they drag a sister chromatid to one end of the cell or to the other end of the cell, and that works also because there are motor proteins attached at the kinetochore on the sister chromatid that then walk along the microtubule, and so they're dragging the chromosome towards the centrosome at one end of the cell or towards the other end of the cell. So when we've got a pair of sister chromatids and we attach a microtubule, is what we're then going to do is actually pull those sister chromatids apart and put them in each end of the cell. So that's what's going to happen in anaphase. It's the pulling apart of the chromosomes. And so that's anaphase. And then the last phase here, telophase, we're going to come and look at cytokinesis separately, so I'm going to cross that out. So in telophase, what's going to happen is that the chromosomes are going to get to each end of the cell, and then the nuclear envelope is going to start to reform. So here is the nuclear envelope starting to reform, and that's going to happen at opposite ends of the cell. The microtubule spindle starts to break down, and so that's going to go away, and the chromosomes are going to start to decondense, and then this structure called a cleavage furrow. So this is where the cell membrane is like starting to invaginate, and that's the start of cytokinesis. Now that's not actually cytokinesis, but that's just the start of it, which happens at the end of telophase. And so there's this structure which starts to pinch in. So if you think about the cell like this, and then it starts to pinch in, and it kind of gets like this, at that point, that's the start of cytokinesis. So that's a quick run through of the events of mitosis, and you want to be making sure you can recognize the behaviors of the chromosomes, the nuclear envelope, and the centrosomes as you look at this. So let's look at cytokinesis. So just a couple of images to help you see what's going on here. So here is what we've got is the nucleus is reforming here and here. So two new nuclei, and then is what you start to see is you can just make out this ring of actin. So here we're seeing the actin stained in green. So the ring of actin, you've got to imagine it kind of forming a ring in 3 dimensions like this. Think about this being like an oval going back into the screen. And so where the cleavage furrow forms, that ring of actin, working with myosin, which is a motor protein you find in muscle, basically causes the ring to contract. And so if we look at the middle, so if we look through the middle of the cell, we see this ring of muscle. No, it's not muscle, but we'd see this ring of actin and myosin contracting, and so we'd see the cell basically pinch itself into 2. And so here's the cleavage furrow, and we're nearly done with cytokinesis here. And you can see the nucleus is starting to reform here with the DNA stained in blue. And so that cell is now pretty much just about done with cytokinesis. So that's what happens in animal cells, but this also happens obviously in plant cells, and things are a little bit different for cytokinesis in plant cells. So in plant cells is what we're going to do is we're going to take a single cell. So here's a single cell. Let me just outline the single cell. Not very good at outlining things, but you get the point. So there's the single cell, and here is one set of DNA, one set of chromosomes. Here's another set of chromosomes. And the metaphase plate was right here. So that's where all the chromosomes lined up. And so what's going to happen is at the point where the metaphase plate was actually present, remember that's not actually a structure, it's just where all the chromosomes line up in the middle of the cell. But at that kind of imaginary line, if you like, so what's going to happen is lots of pieces of membrane is going to start to fuse together, and that's going to give you a new membrane. And on that new membrane, you build a new cell wall, and that's called the cell plate. So rather than splitting and pulling two new cells apart, is what we're going to do here is just take a box and divide it in two. And then those two boxes will grow, and they can be divided in two. And so we can divide in the plane I've shown you here, or we could also divide in this plane here. And that basically determines whether cells are going to be pushed up or pushed down or sideways, whichever way cells are going to get pushed. Okay, so there is our run through of the events of mitosis and cytokinesis. I'll be back shortly and we'll start looking at meiosis.